Welcome, and thank you for listening to this presentation on targeted cancer therapy, choosing the right assay for PARP inhibitors. I am Veronique Baron, Scientific Applications Manager at BPS Bioscience. And before I start talking about the PARP proteins, let me quickly introduce you to BPS Bioscience. We are a scientist-founded, scientist-driven company with a mission to advance new scientific discoveries that led to therapies by creating innovative solutions for research. The company was established in 2005 by Dr. Henry Zhu. We're innovating drug discovery in immunotherapy, epigenetics, for the last 2 years in SARS-CoV-2 and cell signaling. Industry leader in recombinant proteins, we specialize in hard-to-purify protein complexes. We are leading producer of novel recombinant cell lines and cell-based assays. We are dedicated to delivering novel assay kits, cell systems, and virus-based tools. Our core technologies enable the rapid development of drug discovery tools that we provide as off-the-shelf products and services. We specialize in making protein and conjugates, engineering cell lines for specific research purposes, such as CAR T cells, using CRISPR-Cas9 for gene editing. We specialize also in virus engineering and production, including lentiviruses, VSVs, and adeno-associated viruses. We provide screening and profiling services. And finally, we're currently adding capabilities in induced pluripotent stem cells. Very soon, we will be launching our first product, iPS-derived chemiluminescent. We are ISO certified, so you can trust our quality. The ISO certification is an international standard that specifies requirements for a quality management system. It is built around the concept of organizing processes and continuous improvement in order to meet customer needs. The number of citations of our products in peer-reviewed publications has been growing consistently, indicating we are making an impact on a community of discovery. Now, let's move on to today's topic. The presentation will start with a short introduction on PARP proteins, moving on to an overview to their function, their clinical significance, and the discovery of PARP inhibitors. Then we will take a dive into PARP assay formats and design. I will detail 4 types of assays: colorimetric and chemiluminescent ELISAs, AlphaLISA homogeneous assays, PROTAC optimization assays, and finally, the very unique PARPTrap assays. PARP is a critical enzyme involved in DNA repair and also in many other cellular processes. I will come back to this. It also belongs to a network of over 150 proteins involved in DDR for DNA damage response. PARP is not just one enzyme. It is a family of 17 distinct proteins shown here that differ in size, in structure, in function, and in subcellular localization. You can see here, for example, that PARP can be located in a nucleus, in the cytoplasm, or in both. As you can see on the table, they also differ in enzymatic activity. In particular, they can catalyze either the poly-ribosylation of proteins or the monoribosylation of proteins. Some of them are very particular. PARP4, which is also called VPARP for volt, means that the protein is part of the complex involved in intracellular transport. PARP5s, which are also called tenkirase, TNKS1 for PARP5A and TNKS2 for PARP5B, are involved in telomere maintenance. They contain a very large anchoring repeat, which is very specific for these 2 PARPs. Some of them contain zinc fingers, some of them contain regulatory domains, macrodomains. Only PARP1, 2, and 3 are DNA dependent. PARP1 and 2 are currently the most relevant to oncology and the best studied PARP factors. Now, PARP proteins use NAD+ to append ADP ribose units either on themselves, which is called ribosylation. This would be the case here, for example, if the protein substrate is PARP itself, or on various target proteins. And these proteins can include histones, topoisomerases, DNA polymerases, DNA ligases, and so forth. This is the NAD+ here, and the part that I have circled is the part that is appended to the protein with the release of a nicotinamide. Now, ADP ribosylation comes in several forms. As I have mentioned, some PARPs are capable only of mono ADP ribosylation, which is called methylation. Others are capable of poly-ADP ribosylation, which is called PARylation. And a PAR is a polymer that can contain up to 200 units. So I've shown here a mono ribosylation, a linear polyribosylation, and then a branch polyribosylation. And you can see that there could be different branches happening here, branching about every 20 to 50 units. They're usually attached to a glutamic acid or an aspartic acid or the lysine, and it is a covalent modification. Now you can also see that every unit adds a negative charge to the target. So the branch PAR-related entity, for example, is going to be much more negatively charged than a mono MER-related entity. I have indicated here some of the consequences of ADP ribosylation on protein function. So if the ribosylation happens on histones, you could have chromatin relaxation. It can alter the activity of DNA synthesis proteins such as polymerases and ligases and so forth. It will alter the activity of DNA repair proteins, and it will alter transcription. It regulates cell cycle by ribosylation of FOS, pCNA, and more. It can be a trigger for the recruitment of other proteins, resulting in the formation of protein complexes. PARylation can inhibit enzymatic activity. It can result in steric hindrance and in functional inhibition. And it can result in the repulsion of negatively charged molecules, such as DNA, for example. Now, this slide shows how PARP proteins have pro-survival or death functions. It's a complicated slide, and I was not going to go into details for this, but I wanted to emphasize the diverse functionalities of the family members. So PARP1 here is involved in the death signal of trial receptors, for example. PARP5s are involved in PTEN signaling. PARP13 here is involved at the level of the exonuclear complex. PARP16 is involved with chaperones, the antioxidant response, the unfolded protein degradation. PARP14 is involved as part of JNK kinase pathway. PARP2 is in the nucleus, and you can see that PARP1 is involved in many different functions at the DNA level. Now, this one is a very simplified diagram of the role of PARP1 and 2 in DNA damage. So PARP translocates to the nucleus due to a DNA damage. It will use NAD+ for polyPARylation of itself and of other proteins, and this recruits repair proteins to the damaged DNA. Then the DNA is repaired, but PARP itself, because it becomes so highly negatively charged, will dissociate from the DNA. And then the PAR-related branches are removed by a protein which is called PARG. There is a release of the ADP ribose monomers, and the free ADP ribose exits the nucleus, can be used as a signaling molecule. And this exemplifies how PARP1 and 2 play a key role in maintaining genome integrity. At a higher level, this diagram represents the wide range of functions carried out by the PARP enzymes. Chromatin remodeling, where pyrylation determines conformational changes of the chromatin. Methylation, for example, PARP1 neutralizes the activity of DNMT1. It's involved in RNA biology. For example, the promoter RNA mediates the association of TIP5 and PARP1 and activates PARP1 to pyrylate itself, TIP5 or histones. Other emerging interactions of PARP1 with some nuclear proteins, other crosstalks are being studied right now, and quite recently it's been shown that PARP is involved in response to infection, and that the consumption of NAD in cells is dramatically increased by activation of PARP1. So that was the case, for example, for SARS-CoV-2 and COVID-19 disease. As I mentioned, PARP1 and 2 are involved in DNA repair, and at present this is the most significant in oncology because DNA damage really is pervasive and has been directly linked to the formation of tumors. So DNA damage is caused by ultraviolet or ionizing radiation, by chemicals, chemotherapy, reactive oxygen species, by normal cell metabolism, or by proliferation. There can be up to 10,000 single breaks per cell per day, and up to 10 to the 17 DNA lesions every 1 to 3 days in the human body. Reactive oxygen species, metabolism, and cell proliferation are all increased and accelerated in cancer cells. So it can be easy to see why PARP1 and 2 would be involved in tumors. It's also been shown that defects in DNA damage pathways are often selected for. And conversely, genetic alteration of DNA repair genes is directly known to cause cancer. The first PARP inhibitors were discovered through synthetic lethality. So what is synthetic lethality? It's when the simultaneous alteration of two proteins causes cell death, but the alteration of either one does not. This is a concept that I have represented here. So say, for example, that we're studying PARP and another DNA damage response protein called protein B. When both of them are working normally, the cell is very happy and will live. When one of them is altered, so for example, if there's a genetic mutation in the gene encoding protein B, the cell can still repair the DNA because PARP is not altered, so the cell will still live. If PARP is inhibited but protein B is there is no consequence. However, if there's a mutation on gene B and you use PARP inhibitors in these cells, you will find that the cells cannot repair the DNA anymore and they are going to die. So that was the case, for example, for cells deficient in the genes encoding BRCA1 or BRCA2, which would be protein B in this case. And it turned out that PARP inhibitors in these mutated cells were very efficient at killing the tumor cells. PARP1 and PARP2 are the most studied therapeutic targets of the PARP family. Their inhibitors are currently approved in ovarian cancer, triple-negative breast cancer, and prostate cancer, and there are many more inhibitors in development. Other members of the PARP family are of interest as well. For example, PARP7 is involved in interferon signaling and in the immune system. It's become a therapeutic target in NASH, non-alcoholic steatosis hepatitis. PARP3 is involved in oxidative stress and in neurodevelopment. It is a therapeutic target in cancer. PARP6 is involved in spindle formation and is a therapeutic target in various types of cancer at the moment. There are now 4 FDA-approved PARP inhibitors: olaparib, niraparib, talazoparib and rucaparib. I have indicated their brand names right here, as well as previous alternate names, the company that produced these inhibitors, and their structures. The first was approved by the FDA in 2014, that was olaparib, and rucaparib was approved in 2018. So if there are PARP inhibitors on the market, is there a future for new developers? Well, yes, absolutely, which is why there are hundreds of clinical trials for new inhibitors or for new applications of the existing ones. So for example, we need new drugs with lesser off-target cytotoxicity or better pharmacological properties or better solubility. New indications could be found for these inhibitors. For example, an area of study currently is about the protective effects of some of the PARP inhibitors, for example, the neuroprotective effects. There are many types of cancer that could be treated with a PARP inhibitor, and this is still an area in development. Other biomarker mutations have been found. So for example, it is possible that ATR mutations could open the door for new PARP inhibitors. Combination therapies, combining PARP inhibitors with chemotherapy or radiation, is synergistic in many preclinical models. So there are many new indications that we could find here for PARP inhibitors. In addition, we need to overcome the resistance that has been observed after treatment with some of the inhibitors. So it's been shown that partial function of BRCA2 is restored by reversion of the initial mutation. So second generation, third generation PARP inhibitors may be needed. In addition, other PARP members have distant functions that can be involved in diseases, and so these new PARP members also need corresponding inhibitors. Which leads us to the topic of designing biochemical assays for drug discovery and development. There are several types of assays that have been designed to screen or characterize PARP inhibitors. Each assay has its own advantages. So I have summarized here some of these advantages. Chemiluminescent assays and colorimetric assays are AlphaLISA type assays, so they're not homogeneous, they will require washes, there are more steps, so they are slower to complete. But they also have a very low cost. They're not as amenable to high throughput studies, which is why TR-FRET, fluorescence polarization, and AlphaLISA may be preferred for screening, for example, screening small molecule libraries. These are homogeneous assays that do not require wash steps. Therefore, they are fast to completion. They're more amenable to high throughput, and they allow for very small volumes. However, their cost is significantly higher than chemiluminescent and colorimetric assays. So all these are factors to consider when you're choosing for a kit, but also your ultimate goal and what you're trying to do is going to determine what kind of assay format you will use. These are some of the technical aspects of designing a biochemical assay. There are various parameters to optimize. So the enzyme concentration needs to be optimized, the buffer composition, the substrate needs to be chosen carefully. There are PARP-specific substrates that allow better measurement of the specific activity. For example, PARP1 favors 3' phosphorylated double-strand break termini or DNA mimicking double-strand breaks, and some single-strand breaks, but PARP2 does not. You may also consider whether your favorite PARP catalyzes pyrylation versus methylation. It can influence the time of the assay. It can influence the intensity of the signal, and of course the assay sensitivity that will result. Not all PARPs are DNA dependent. So PARP1, 2, 3 are DNA dependent, and for the assay to work, you will have to find the optimal DNA probe that needs to be added to the assay for it to work. So the conclusion of this is that there are unique assay conditions for each of the PARP family members, and you will need to design the assay for every PARP that you want to study. One critical parameter is the quality of the protein used in the assay. So here's an example of recombinant PARP1 purification. Here, the protein is engineered with a tag to allow for affinity purification. And this is a Kumasi staining on the SDS page to visualize that there is just one band on the gel and that it is highly pure. Of course, the purification process should not alter the enzymatic activity of the protein. So if you're going to perform multiple assays, there should be a good quality control process to ensure the reproducibility from one assay plate to the other. In this case, we have titrated enzymatic activity, and we can compare one lot to the other and make sure that they're very reproducible. This is an example of a dose response olaparib inhibitor using assay kit 80551. Now let's talk a little bit about colorimetric and chemiluminescent assay kits. I will describe the principle of the assay, which is an ELISA, and it measures the enzymatic activity of the protein. I will show example data and a couple of case studies taken from of the products. So this is the principle of this assay. You add histone to a well, and histone is going to coat the bottom of your well. Then you add your PARP of interest, NAD, and biotin NAD, and you wait for ribosylation to occur. You add a streptavidin HRP, which is going to bind the biotin in the NAD. You add an ECR reagent, and light is produced. So this is for the chemiluminescent kit. If this were a colorimetric kit, you would add a colorimetric substrate of HRP. That means enzyme-linked immunosorbent assay. It is not a homogeneous assay. You will need several wash steps, and it is a slower procedure than homogeneous assays. It directly measures PARP enzymatic activity, either parylation or methylation. So this here is a show of parylation, but we also have assays that can be designed with a single ribosylation, monoribosylation. So this here measures the incorporation of the ADP ribose into a histone-biased substrate. So the signal is directly proportional to the enzymatic activity of the PARP. It is an inexpensive assay. The signal can be very stable if you're using the colorimetric version of it. However, sensitivity is much better using the chemiluminescent version of it. So this is an example data in which we have used colorimetric or chemiluminescent kits. We have these kits for most PARP proteins, and we can compare the IC50 of a compound against pretty much all available family members to determine the specificity or selectivity of a drug. In this example, we have used olaparib and AZD5305 against PARP1 and PARP2. And what we see is that these two inhibitors have the same affinity for PARP1, but they don't have the same affinity for PARP2. We have about 2 logs difference between olaparib and the AZD compound here against PARP2. That indicates that the assay is sensitive and selective for PARP1 and PARP2 if we can see such differences. We can measure the IC50 of compounds against PARP7 or against PARP11 and other PARPs as well. Now what I'm showing here is a case study where the authors studied a PARP radiotracer for PET, positron emission tomography. PET is a sensitive, non-invasive, real-time measurement of target engagement in vivo. Therefore, it can provide a lot of information on pharmacokinetics and dynamics. It usually uses a radio-labeled PARP inhibitor with a fluorine 18 isotope, which is the most widely used isotope. And currently, there are analogs of olaparib and rucaparib that are tested in clinical trials, where you can see where the inhibitor is going into the body and what tissues are affected and whether the inhibition is efficient. So in this study, the goal was to look at the usefulness of an F18 analog of AZD2461, which has different affinities for different PARP members. And the study was to uncover the significance of their selectivity profile when performing PET imaging. So it's a study from Gubal et al. Published in 2020. So what I'm showing here is their PARP binding profile for the AZD analogs. I'm showing the structure of each analog. This is the AZD. This is the proto-devorylated analog here in the middle. And this is their positive control for the study, which is olaparib. And they used the chemiluminescent PARP1, 2, 3 assay kits to explore the binding of the F18 labeled PARP inhibitors. So we can see here the various titrations that they used for drugs. In this study, Kinozada et al. Were studying a novel telomerase inhibitor, which is called K476. They identified this inhibitor which blocks the immunosuppressive beta-catenin pathway, and they observed that this inhibitor enhances the anti-tumor effect of anti-PD-L1. They used our kits to look at the selectivity of the inhibitor across different family members. So what we can see here is that used at 3 different concentrations. The inhibitor inhibits PARP5, the telomerase, much better at 100% efficacy, much better than other PARP family members like PARP1 to 3 or PARP6 to 12. Only at the highest concentration did they see significant inhibition of PARP11, PARP12, and other PARP family members. So that indicates that at these concentrations, the TNKS inhibitor is actually pretty selective for the telomerase. Let's move on now to the AlphaLISA homogeneous assay kits. I will talk about the principle of the assay that also measures PARP enzymatic activity, and I will show examples for these assays. So this is the principle. It's an in-solution assay based on the AlphaLISA from Perkin-Elmer. It directly measures PARP enzymatic activity, either pyrylation or methylation, by measuring the incorporation of ADP ribose into a histone-based substrate. So this is all in solution. Nothing needs to be attached to the well. You have a biotinylated histone substrate, and then you add to it either TNKS or PARPs and NAD+. So you trigger the pyrylation of the histone or the methylation of the histone. Then you add an ADP ribose binding reagent with acceptor beads. So this reagent is going to bind to the ADP ribose, and the acceptor bead is going to bind to the binding reagent. This is shown here. And then you add donor beads that are streptavidin labeled and are going to bind to the biotin. So this here allow the transfer of energy between the donor bead and the acceptor bead, which will trigger the signal. And of course, this can happen only when the donor and the acceptor are close enough, which can happen only when all the components are there and the histone has been ribosylated. So it's a very amenable to a high throughput format. It accepts very small volumes. It can be done in 384 well plates. It has a very broad dynamic range, and it's a fast assay, so you can really perform the assay in just a couple hours, basically. This is an example data obtained with this type of assays. So you can screen a small molecule library. You can compare the IC50 of a compound between family members, and we have these assays for PARP1, 2, 3, for the tankerase, both of them, and for PARP11. So the PARP1 homogeneous assay was tested with olaparib, talazoparib, and AZD5350. We can see that they pretty much all have the same affinity for PARP1 in this type of assay, the same IC50, I should say. And we also tested olaparib and talazoparib in the PARP3 homogeneous assay. Now let's talk a little bit about the PROTAC optimization kit for PARP1 cereblon binding. So I will first remind the audience what PROTAC is. I will explain the principle of this PARP assay, and I will show a couple of example data. So what is PROTAC? PROTAC stands for proteolysis targeting chimera that causes protein degradation by the targeted recruitment of a ubiquitin E3 ligase to the protein of interest. So here is your target protein, and you want it to be degraded by an E3 ligase. You will add an E3 ligase ligand linked to a ligand for the protein of interest. Now, this here requires a lot of optimization. You need something that can bind to your E3 of interest, another moiety that can bind to the target protein, and you need to optimize the linker very carefully to make sure that both retain a good affinity, good selectivity for the target and for E3, can put them together so that the target protein can be degraded. So once your PROTAC is optimized, it brings the target protein together with the E3. The target protein is ubiquitinated, recognized by the protein, and is degraded. The advantage of this system is that in theory, any protein of interest may be targeted, even proteins that are deemed undruggable and proteins that are very difficult to target. As I said, it requires several steps of optimization, and this can be very difficult or time-consuming. So this assay really was designed to simplify the optimization of a new PROTAC of interest. In this case, the protein targeted is PARP1, and the E3 ligase is cereblon. So this assay can be used to screen for new moieties that bind PARP1 or for new moieties that bind to cereblon and optimize the linker. It is an in-solution assay, so it is homogeneous and very rapid. It measures the formation of this complex, which is mediated by the PROTAC of interest. It is homogeneous, high throughput. It has a very broad dynamic range and is very fast. So in this experiment, the PROTAC of interest, which is a positive control, irucaparib AP6, interacts with both PARP1 and cereblon, bringing them in close proximity. PARP1 contains a GST tag, which is recognized by the glutathione donor bead, while the cereblon contains a flag tag that binds to the anti-flag acceptor bead. Upon excitation of the donor bead, you have a singlet oxygen that is generated by the donor bead. It excites the acceptor bead, which emits light proportionally to the level of interaction. This is an example of the data that can be obtained. The applications for the assay can be to compare the activity of different PROTACs. It can be to discover and optimize PROTACs targeting PARP1 and cereblon. We tested the assay with irucaparib AP6, and you can see the difference in signal with and without the PROTAC. Of course, the interaction is mediated by the PROTAC, so the signal increases as you add increasing concentrations of the PROTAC. In this case, the PROTAC-mediated PARP binding to cereblon was measured in the presence of rucaparib and in the presence of polyamidomide, where we see that these inhibitors prevent the binding and the formation of the complex. Now let's talk about PARPTrap assays. We will describe the mechanism of PARP trapping to DNA, talk a little bit about the clinical significance of PARP trapping, what fluorescence polarization is, because the assay that I will describe is based on fluorescence polarization, the principle of this particular assay, and example data. So this is a little diagram of the mechanism of PARP trapping. So when PARP1 or PARP2 bind to a site of damaged DNA right here, it adds poly-ADP ribose chains to its own backbone and to DNA damage repair proteins. And that recruits this protein, activates the protein to the site of damage. The PAR-related PARPs are going to detach from the DNA because they are so heavily negatively charged. That will free the site and will allow the repair process to take place. Now, it's been observed that some PARP inhibitors that compete with NAD+ in the NAD+ binding process will prevent PARylation altogether and will prevent the dissociation from the DNA. So the continuous presence of PARP at the site of damage will prevent repair, will block replication, and will lead to cell death. So drugs that trap PARP1 or PARP2 to the DNA tend to be more cytotoxic than other PARP inhibitors, and that is highly desirable. And the clinical significance of this increased cytotoxicity cannot be understated. The capacity of a drug to trap PARP on a DNA is usually correlated with cytotoxicity, and this is a cartoon of what is known of the existing drugs. So for example, talazoparib has a very high capacity to trap PARP. It's also the more cytotoxic among these drugs. Niraparib is a little higher in its PARP trapping efficacy than olaparib and rucaparib, and the least efficacious to trap PARP is veliparib. So our assay for PARP trapping is based on fluorescence polarization. Fluorescence polarization is a very complex technique, and if you're not familiar with its principles, I invite you to look at our ebook in our resources section of the website. This ebook explains all the technical principles behind a fluorescence polarization assay. Briefly, the technique takes advantage of the fact that when a small fluorophore is excited by a polarized light, which means that the light is processed through a polarizer before it hits the fluorophore, the light becomes depolarized at emission because the small fluorophore rotates fast because it's small. So that results in low fluorescence polarization. When the fluorophore binds to a larger partner, the molecular rotation slows down because of the large size, and the emission remains mostly polarized. This results in high fluorescence polarization. And it is the difference between these two states, the difference in the fluorescence polarization that is emitted, that's measured in a fluorescence polarization assay. So in this PARPTrap assay, a small piece of DNA is labeled with a fluorophore. It is small, therefore it results in low fluorescence polarization when it is excited. Binding of PARP1-2 to this little piece of DNA means that it's now a much larger entity, and that results in high fluorescence polarization. Adding NAD to the reaction triggers PARylation of PARP, which becomes highly charged and dissociates from the DNA, and this results in low polarization. Whereas if you have a PARP inhibitor in the reaction that binds into the NAD binding pocket, you don't have PARylation anymore. PARP is trapped to the DNA, and the fluorescence polarization remains high. So the difference in polarization between the initial state and the final state is indicative of PARP trapping. And the increase in fluorescence polarization from start to finish is directly proportional to the amount of the PARP trapping. So this is an example data and a proof of principle titration of known PARP trapping inhibitors. We have tried talazoparib in red, AZD in green, olaparib in blue, and veriparib in black. And what you can see here is that the relative trapping efficacies of talazoparib, olaparib, and veriparib are very similar to results that I have shown in 2 or 3 slides prior. Now what we can also see is that talazoparib, olaparib, and veriparib have similar trapping efficacies against PARP1 or PARP2 as measured by the EC50. But AZD was 2,000 times more efficient against PARP1 compared to PARP2. This is the green line right here. And that demonstrates great selectivity of this compound between PARP1 and PARP2. AZD305 displays similar DNA trapping activity to our PARP1 as the best in class, which is talazoparib. Now to finish this presentation, I will point out that we offer custom services, either custom protein design and production, custom assay design. We can test drugs on any one of the PARPs that we have available, whether it's a dose response or single doses, and we can compare drug efficacy across family members. We have most PARP family members available to assay, and we can demonstrate that a drug of interest has better affinity for a target or another. These assays are available for chemiluminescent format, colorimetric, homogeneous, PARPTraps, and we also have an inhibitor set if you wanted to compare different inhibitors against the same PARP. So in this case study, Carney et al. Were measuring target engagement of PARP inhibitors in small cell lung cancer. They are using non-invasive quantitative assessment of dynamic intratumoral target inhibition using 18F PARP inhibitors and PARPi-FL, which are 2 currently used PARP inhibitors for PET imaging. And I think that this may improve patient care through real-time monitoring of drug delivery. This is a little example of what can be done here in the mouse, for example. So in this study, they asked us to perform PARP enzyme family binding assays using the colorimetric assays with a histone substrate, biotinylated, labeled NAD at 100 nanomolar here, 50 nanomolar in the middle, and 10 nanomolar on the right for each inhibitor and for each enzyme that they were using. So this here is the chemical structure of their different compounds, and we can see that we could measure the selectivity of these different drugs for the various PARP members. These are example IC50 determinations in that same study where we compare PARP1 here and PARP2. In summary, we have seen that PARP proteins play critical functions in the cellular response to DNA damage and in many other biological responses. Several PARP inhibitors have shown great efficacy in tumors with defects in their response to DNA damage. New inhibitors and new treatment modalities are under investigation. A variety of PARP assays exist to reach various experimental goals, allowing diverse approaches to PARP targeting. Thank you for listening.